Hello and welcome back to Microbiology. Today we are going to talk about some modern implications of microbiology, some more modern things that have happened, and we'll start with talking about chemotherapy. So when you hear that word chemotherapy, often that just evokes cancer, but chemotherapy actually means any therapy with any chemical. So anytime we're using any chemical to treat an illness. And the reason that works is because a lot of chemicals are going to kill microbes faster than they're going to kill people. So it's more toxic to that microbe than it is to the person. This includes two different categories of drugs. Although in the real world we rarely differentiate this, it's kind of nice to know some drugs are synthetic. That means that it is made in a lab, artificially created by people from other chemicals. And antibiotics are chemicals that bacteria or fungi make. So living cells, living microbes make these chemicals. And these are chemicals that will inhibit or kill other microbes. So it's a way that bacteria and fungi have evolved a way to prevent other bacteria or other microbes from encroaching on their space, kind of gives them a benefit to grow. But we're able to take those and use them to kill other microbes that we don't want around. The first synthetic drugs involved quinine, which was taken from a tree bark, and it was used to treat malaria. Paul Ehrlich, though, was a scientist who wanted to find a magic bullet. And this magic bullet would destroy all infections and never harm any people. So if you think about it, he was working in the early 1900s. So he's had over 100 years that science has had. Can you think of anything that qualifies as a magic bullet that kills any pathogen but doesn't hurt people? So you probably can't think of anything because there isn't any such thing. But he did develop this drug, salvarsan. So salvarsan was a drug that treated syphilis. It was based on arsenic and it would kill the syphilis bacteria. Hugely important because syphilis was, up until that point, it could develop into a hugely debilitating, horrible disease. So being able to treat that illness was very important. By the 1930s, sulfonamides were synthesized. These are drugs that are still widely used. A lot of sulfa drugs. If you've had a drug that has sulfa something in it, it may be a sulfonamide. In 1928, Alexander Fleming discovered this antibiotic penicillin. So he had been doing some research on this bacteria, Staphylococcus aureus, and he had some plates that he went out for the weekend and he just forgot to stick them in the refrigerator. He just forgot about them. And when he came back in, he saw one of them had mold on it. And instead of just being like, oh, that stinks, and throwing it away, he stopped and paid attention. And if you look, here, this is the mold colony. When you're really close to the mold, you don't really see a lot of bacteria growing. And the ones that are just really tiny, not really normal shaped. This is what those colonies should look like a nice, big, fried egg spot. You don't see that. You just see barely anything going on here. So we said maybe something about this mold, something about this penicillium is inhibiting the growth of my bacteria. At that time, it took a while until penicillin, the antibiotic that was taken from penicillium, it was not clinically tested until World War II when we were wanting to save our soldiers on the battlefield. But very quickly after that, penicillin became available for mass production for people to use to treat all kinds of bacterial infections. However, there are some problems with using antimicrobial chemicals. One is that overuse can lead to resistance. So overuse of antimicrobials can lead to resistance. For example, with penicillin, it started being widely available in the 1940s and 1950s, and it wasn't long until there was penicillin resistance. Some drugs can be toxic to humans. So some drugs you can only take short term. You can only take in small amounts. If you take too much or for too long, it can cause damage. It could damage your kidneys. It might damage your hearing. It might damage how your teeth form. There's different ways that different drugs can be toxic. And so research to figure out how to overcome these problems is what ushered in the third golden age of microbiology. This antimicrobial resistance is a big problem of research that is still currently going on. So, modern microbiology is divided into a lot of different, more specific forms of study. So, this ending ology tells you you're studying something. So, bacteriology is studying bacteria. Mycology, anything with myco means fungi. So, mycology is studying fungi. Virology is studying viruses. Parasitology is studying parasites. Often that means parasitic worms and protozoans. Genomics is studying the genes. So see this root word genes. Studying the genes of an organism. This provided a great new way to classify organisms. Instead of just classifying them based on what they looked like, what shape they had, we could actually see how closely their genes were related. Microbial genetics is studying the genetics or the genes specifically of a microbe, how microbes inherit their traits. And molecular biology is studying how DNA directs those cells to produce proteins and to have the different traits and functions that they have. DNA really has not been very long that we've really understood what it was doing. In the 1940s, it was Avery, McLeod, and McCarty that discovered DNA was a hereditary material. Before that, we didn't really know, was DNA the hereditary material or was it proteins? How were you inheriting traits from your parents? So Avery, McLeod, and McCarty finally proved that DNA was being passed on. In 1953, Watson and Crick published a paper showing the structure and replication of DNA. Although the paper was really based on work that they stole from Wilkins and Franklin. So really it's Maurice Wilkins and Rosalind Franklin who get credit for this, for understanding the structure and replication of DNA. So how DNA is shaped, how it's replicated, how it copies itself, how it can be passed on to children. Paul Berg in the 1960s created recombinant DNA. So this is really a big step at this point. So you can see combine here in the middle of this word recombinant because we are combining DNA. We're taking DNA from two different sources. Particularly, you can take a gene from an animal and stick it into a bacteria's DNA, and that bacteria will start producing that protein from an animal. We do this a lot for insulin to treat diabetics. We can take a gene for insulin and program it into a bacterial cell DNA, and the bacteria will start creating insulin so that we can provide insulin to people who don't create their own. Microbial ecology is studying how microbes affect the environment. One important thing that microbes do is they take elements in forms that we can't use and turn them into forms that we can use. So they take carbon dioxide and create sugars from it. They take nitrogen in the air and create ammonias and amino acids from it. Ways that we would never be able to access them, we can start accessing these. We would never have nitrogen available to plants and animals without bacteria to take the nitrogen from the air, to fix that nitrogen. Bioremediation is using microbes to remove some sort of pollutant. We can take bacteria and program in a gene to give them the ability to digest oil so they can start degrading an oil spill. They can break down sewage. They naturally degrade sewage in most cases anyway. Here you have people spraying bacteria onto some sludge. It's probably a pile of sewage. They can clean up a mercury spill. Mercury is also very toxic to people, but bacteria can be used to remediate or to clean up that environment. Biotechnology is using microbes to make foods and chemicals. We discussed this in the part 1 of the chapter 1 lecture. Things like cheese, beer, bread, vinegar, all created by microbes. And then recombinant DNA is using bacteria and fungi to produce proteins, to produce vaccines, to produce enzymes. So we can create some sort of gene therapy to replace or repair a missing or defective genes. We can use modified bacteria to protect crops and insects from freezing. Microbes can be used as biological insecticides. So there are some microbes that can kill insects without harming people, and they don't kill the food, they don't kill agricultural crops. So this man is spraying this bacteria called BT, or Bacillus thuringiensis. He's spraying this onto the crops. It won't affect the crops will grow just fine. It won't affect people who eat this, people will be just fine. But any insects that come along and try to start eating this crops will die. So your crops will not be infected with aphids or caterpillars or any other bugs that come and eat it. It won't stop a rabbit from getting in there and eating it, but it'll at least stop the insects. Okay, so at this point, you should be able to describe to me Ehrlich's magic bullet. You can describe to me bacteriology, mycology, parasitology, immunology, and virology. You can differentiate between microbial genetics, molecular biology, and genomics. You can describe beneficial uses of bacteria. You can differentiate biotechnology from recombinant DNA technology. I'd also like you to be very comfortable describing what Pasteur did and be very comfortable describing how we got penicillin. Okay, I will see you back for our next lecture video where we will discuss some emerging infectious diseases. See you later.